So welcome everyone. My name is Paulina Magaña and I'm part of the AlphaFold database team. I'll be presenting today's webinar on accessing and interpreting predicted protein structures from the AlphaFold database. The objectives of today's webinar are first gonna talk a little bit about what proteins are and what the protein folding problem is. Then how AlphaFold works, what's AlphaFold, and how we can interpret then the predicted structures that we get from the algorithm. Then I'm going to just talk briefly about the impact of AlphaFold, and for the last thing, I'm going to just cover how you can access the predicted structures from the database. First of all, a little bit of background on what proteins are. Well, proteins, they perform a wide variety of functions, such as catalyzing biochemical reactions, transmitting signals between cells, and they also provide structural support. They are made up of long chains of smaller molecules called amino acids, and the sequence of these amino acids is what determines the unique shape, the 3D shape, and the function of each protein. So the exact 3D shape is important for the protein's function, and if we can understand the structure, then we will be able to get an insight into the functions. But there's a gap between the protein sequences that we have and the available structures. So in UniProt, we have over 250 million protein sequences. But if you look at the available structures in the PDB, you can see that we only have over 210,000 entries. And if you try to see what this is for unique proteins, it corresponds to almost 63,000 unique proteins. So we are not yet close to close this gap, and it has grew bigger within the last decade. So the predicted structures will be able to help closing the gap that we have between the sequences and structures. But what's the protein folding problem, and why do we have less structures than sequences? So this is just to get a visual idea of the steps involved in the structure determination, from the DNA cloning to purification to the determination of the structure. But there are so many troubleshooting steps in every part of this diagram, and it's very time-consuming. So experimentally techniques for getting these structures are laborious, and they usually take years. So that's the reason why we have a small number of structures that have been determined compared to the sequences, and this is a major limitation for research. So it has been long since postulated that theoretically, if you have the amino acid sequence of a protein, you should have enough information to be able to predict its 3D shape. So when a protein is synthesized, it must fold correctly to carry out its biological function effectively. But then we have Lewinthal's paradox, which highlights that if a protein were to randomly explore all the different conformations that it can have before it finds its native structure, it will require an incomprehensible amount of time to try them all one by one. But still, the proteins fold rapidly and with remarkable accuracy. So what this means, in the context of protein structure prediction, then this dogma and paradox serves as a foundational concept that inspired computational methods that aim to predict the structure of the proteins based on the amino acid sequences as a blueprint. and it highlights the need for an efficient algorithm that can swiftly identify the most likely native structure based on the sequence. So this is where Google DeepMind's AlphaFold comes in play. AlphaFold is an advanced deep learning algorithm that was developed by Google DeepMind. There is an assessment that happens every 2 years. This is a blind test competition where computational biologists try to predict the structure of several proteins that are not yet public. So in 2020, for CAF14, AlphaFold was recognized as a breakthrough with high predicted protein structures. And then on the competition's preferred measure of accuracy, the GDT, this is the global distance test, AlphaFold achieved a median score of 92. And then the algorithm was used to predict the whole human proteome after this. But then what's AlphaFold and how does it work? How do you get your structure? First of all, I'm just going to give you, before I start, I'm just going to give you a high-level overview of how it works. So AlphaFold uses the input sequence to query several databases and then constructs a multiple alignment sequence, MSA. So this MSA is a key input because it contains all the sequences that are evolutionarily related to the sequence target, to your input sequence. But why is this so important for AlphaFold? Well, this allows the discovery of pairs of amino acids that co-evolve. So for protein coding genes, when a residue mutates, there is a compensatory mutation that follows. So then if you analyze the MSA of homologous protein sequences, you can measure the coupling of any given residue path. So for proteins that have no structure, this coevolution measurement could be used as a predictor of which residues are likely to be in contact. So that's the reason why the MSA is important for AlphaFold. And then the input sequence is used to create an array of numbers that represent all the residue paths. Lastly, AlphaFold can use templates from structure templates from the PDB. However, it often produces accurate structures without a template. Although it is most useful when the MSA is small, that's when you might want to add a template to AlphaFold. So in the second part of the diagram, AlphaFold then takes the MSA and the pair representations and passes them through the neural network called EMBL-EBI. So this is extracting information about the relationship between the residues. And the objective of this part is to refine both the MSA and the pair representations. And it's also exchanging information between them. So by the end of this process, the pair representation and the first row of the MSA, they have been repeatedly updated in 48 blocks. And then now contain a lot of information about the structure. So this is then taken to the last part of the diagram, to the structure module. This generates a static final structure in a single step. So what's really important about AlphaFold here is that by feeding back the MSA, the pair representation, and the 3D structure back to the network again, it improves the performance of the final prediction, and it's doing this 3 times. So after the first recycling into the next recycling, it will have a much better starting point than just running the whole prediction again. Also, as part of the 3D structure that you get from AlphaFold, you also get to output for the confidence metrics that you can then use to interpret these predictions. The strengths of AlphaFold. Why is AlphaFold so good? Because it can predict the 3D shape of the proteins based only on the amino acid sequence. It's a very good starting point for you to start creating hypotheses using these structures. And they've been widely used as starting models for X-ray crystallography and cryo-EM. Again, it can use templates from the PDB, but even if the template is available, AlphaFold might discard it if there's enough information from the MSA. AlphaFold then produces 3 outputs, the atomic coordinates in PDB and MC files, and the 2 confidence metrics, the PLDT and the PAE. Going first to the predicted atomic coordinates, you get 2 files. You get the PDB format, which now is obsolete, and it will not be supported for new PDB entries. And you also have the MC file format. So this format is the official format for the PDB archive, and it contains much more information than the PDB. Also, the MC file is extensible, so it means that you can add more information to this file because it contains a well-defined dictionary. It also includes the version history, the cross-references to UniProt, and can be extended to have more metadata. Moving then to the first confidence metric, we have the PLDT, which stands for Predicted Local Distance Difference Test, or PLDT. It is stored in the B factor fields of the MCIF and the PDB files. So what's PLDT doing is that it estimates how well the prediction will agree with an experimental structure based on the local distance difference test. That's LDT. Well, this has been around and has been used for assessing models. Roughly speaking, the LDT is the predicted interatomic distances rather than how well the full predicted and the 2 structures align. So in simple words, this is a local measure, and it indicates the confidence in the local structure. The PLDT here is scaled between 0 and 100, and we color-coded it based on the score. So it goes for dark blue when it's very highly confident, to orange when it's low confident in the region. So note also in this model, it can vary greatly in a chain. So it's important to check the confidence when you interpret all the structural features of a protein. Usually, regions with scores between 50 and 70 are low confidence and should be treated with caution. But if you have the regions with PLDT more than 90, this is expected to be modeled to high accuracy. The PLDT is useful for assessing confidence within domains and possible disordered regions. So if you have less than 50, this is a good predictor of intrinsic disorder. However, it's not going to be like this every time. On the other hand, if you have a high PLDT on all the domains, it also doesn't imply that AlphaFold is confident in the relative position of these domains, because this will require a different metric. So this is why we have the PAE, the predicted align error. This is pairwise and has a value for each residue pair is measured in angstroms. And we use different shades of green to measure the expected position error. It goes from dark green when it's confident to white where the distance is more than 30 angstroms. So this is the prediction of its position error as at a residue X if the predicted and the true structures were aligned on residue Y. And we displayed this as a heat map, and it's a 2D plot. So we're going to see it in action in the next couple of slides. So the heat map that we have on the website is interactive. So you can click and drag a box on the PAE viewer to select the regions of the structure and highlight them on the 3D viewer. So let's see it now. In this case, the 3 squares in the diagonal correspond to the 3 predicted domains. And AlphaFold is highly confident in the packing as this is dark green. But then how can you interpret the relative position of these 3 domains? For that, you also going to need the PAE. So if the PAE is dark green for the residue pairs from 2 different domains, it will indicate that AlphaFold predicts a well-defined relative position and orientations for them. But if the PAE looks white for this residue pairs, then AlphaFold is uncertain and the structure should not be interpreted as in the relative position and or orientations might be wrong. In this case that we see here for this specific protein, AlphaFold is confident in the relative position of these 2 domains, whereas it's not so confident in the position of those 2 domains in relation to the 3rd domain that we see here. We try to highlight this. It's important for you to use both outputs together, the PLDT and the PAE. So here we have an example. This is exporting 4 protein from Arabidopsis thaliana, which is involved in protein export from the nucleus. This protein is predicted to adopt a donut-like shape with an N-terminal helix inside the hole of the donut. And just by looking at it, you might start speculating about whether this helix has a function and there's a reason why it's located there. But it's important to assess the predicted quality and the reliability of the model before making any such inferences. So let's look at the metrics. This helix inside the donut has a very low confidence, as you can see for the orange color, and hence you can't infer that the structure is well protected for this specific region. And then you see the PAE, and it highlights that the position of this helix, which with respect to the rest of the protein, is predicted with very low confidence. So this is just a coincidence that AlphaFold placed it here. Though even if the helix had been well predicted with a high confidence for the PLDT, its location and orientation inside the donut will not be reliable according to the PAE. So as many tools, AlphaFold has limitations. It can only accept 20 standard amino acids as the input. By default, when you run the whole algorithm for predictions, it will give you 5 models per run, although these models used to be quite similar, so means that AlphaFold can't predict conformational variability for a protein. Though we've seen that researchers have come with ways to trick and force AlphaFold into producing different conformations of a protein. A way that you can do this is to provide shallow or a small MSA. So AlphaFold was not trained to predict assemblies. However, we have AlphaFold Multima. It has not been trained or validated for predicting the effect of mutations, although now we have Alpha Missense that has been recently released for variants, and it classifies them as pathogenic or benign. Ligands are not included in AlphaFold, and it cannot make any predictions about any of the non-protein components. It was also not trained to predict structures of nucleic acids. So which protein are we looking at here? This is to highlight the limitations of AlphaFold when predicting different conformations of a protein. Here we see that when exokinase phosphorylates 6-carbon sugars, it toggles between conformational states. So on the left side, you can see the different conformations that it adopts when the sugar is when there's sugar bound, and when it's sugar-free in green. Then on the right-hand side, here we superpose the structure predicted from AlphaFold, and according to the RMSD, which is the root mean squared deviation, we can see that actually the predicted model is the sugar-free conformation in this case. Moving on to the impact of AlphaFold in-house and how this has been used. It's been used to understand disease resistance in bees. So vitellogenin is a protein that you can find in pretty much any animal that lays eggs, and it supports reproduction and can bind to pathogens proteins like an antibody would, and contributes then to the immunity and that is passed on in egg-laying species. Although until recently, little was known about the structure of the honeybee version, which is a hard protein to map, and the main reasons because it's very large, as you can see here. So the predicted structure can be used to visualize its function-specific domains and see how they interact, and then make predictions about their different functions based on the structure that was revealed by AlphaFold. It's been also used to explore the evolution of a protein family. In this case, is the potassium channel tetramerization domain, KCTD. These proteins are linked to a range of diseases, and going again that the structure relates to function. But then little was known about the structural information about these proteins, and using AlphaFold, researchers were able to build a new evolutionary family tree based on the shape of these proteins rather than their genetic sequence. So then how can we access these predicted structures? There are three main ways that you can access the structures from AlphaFold. The first one is you can just download the source code, which is open access, and it is hosted on GitHub. It gives you total control to predict your own protein. It contains all the dependencies and is provided in a container, but you still need to download the genetics and the template databases to run it. One of the good things about this is that you can run your own sequence of interest, you can run large number of predictions, and you have the full freedom to edit the code and tune how the predictions are generated. On the other side, It requires a lot of space for the databases, and you need some coding expertise, and it takes time to run. So there's another alternative to this. You can use Google Colab, which is a Jupyter notebook hosted on Google. This is more limited as how you can tune the predictions, though it's user-friendlier than the source code, and you don't need your local computer to run it. It has pre-written Python scripts, so everything is ready for you to run it, and it executes in the cloud. The good thing is you can run your own predictions. You don't need to know how to code. You can just give your sequence as an input and just run it. On the other hand, it's not the best when you have large prediction jobs. It takes time to run, and is limited on how you can influence the predictions. Something more user-friendly. Well, we have the AlphaFold database. All the predicted structures were made available here. This is a collaboration between Google DeepMind and EMBL-EBI. It was first launched in July 2021. And it now hosts over 214 million predicted protein structures. Good thing is, it's the easiest way to access the predictions. You don't need to code. There are no waiting times, and you can do bulk download from the database. The cons is your protein of interest might be missing in the database. We host the predicted structure, so therefore you're not able to run predictions using the website. I'm gonna cover more about the database and how the different ways that you can access what we have, what we don't have yet. The type of data that we have for all of the proteins, you can find the MC files and the PDB format. You get the two confidence metrics, the PLDT and the PAE. And there's also the metadata. You get the gene name, species name, identifier, and the biological function from UniProt. What we don't yet have in the database is, as noted from the limitations from AlphaFold, the database doesn't contain each structure that a protein, that each possible conformation of a protein of interest. So we don't have that yet. We also don't have viral proteins or isoforms. We don't have assemblings, and there's also no mutant structures or ligands as part of the predicted structures. The database has impacted several fields in the life sciences, in biological sciences, and bioinformatics and computational biology. And it's been cited in more than 2,000 different publications. So this is how the landing page looks. Again, this is a collaboration with DeepMind and EMBL-EBI. And the database provides access to all of the outputs from AlphaFold and the related metadata on a massive scale. Has a basic search system. You can search for your protein using the UniProt ID. The UniProt session, the gene name, the protein name, and or organism. This is the results page, and we highlight every result according to the review status, and if the entries that you have here are part of a reference proteome. We also have a section when you're looking for specific proteins or for the popular organisms in the database and for other that we also host. Every prediction has its own dedicated page. So we provide the data for download to as PDB files, MC files, and the PAE as a JSON file. And you can also provide feedback on the prediction. We also highlight always if there are experimentally determined structures in the PDB, and the biological function of the protein is extracted from UniProt. You can also see the structure straight into the prediction page. We use this 3D molecular graphics viewer called MolStar, and you can use it to measure the distances or change the rendering style. You also have the PAE next to the protein, so it's easier for you to visualize it when you drag a box in the PAE viewer, and then how this highlights the parts of the protein. We are currently working on enhancing the database to include similarity data and functional annotations. So this is hopefully going to come soon. Other ways that you can access the database if you don't want to go and use the website. One of them is the API. We have programmatic access, and it gives you access to all the metadata that we archive in the database, as well as the URLs for the atomic coordinates and PAE. The FTP site is a good option if you want to download large amounts of data. You can make use of it. We also have the BigQuery. For this one, you'll need a Google account and to set up a BigQuery sandbox. And it's a good option if you want to perform complex queries on the data. You want to filter all of the proteins that we have according to an average PLDT, and or using one specific organism. You can also access it through the 3D Beacons API. What's 3D Beacons? 3D Beacons is an open collaboration between the different providers of macromolecular structure models, and you can find here predicted structures as well as experimentally determined. So this is an example of how the JSON looks when you're using the API to query the database. So when you retrieve this is a dictionary that contains the gene name, the entry ID for the AlphaFold structure, the AlphaFold predicted structure. You also see the organism, the UniProt sequence, and the URLs for you to download the coordinate and the PAE. You can also access via FTP. So this is the file transfer protocol area of EMBL-EBI. And this is going to give you access to the current and the previous versions that we have in the database. So at the moment, we host the 4th version. So if you want to see how this has changed, you can just go back to the first version of the database. There's also a README file. So you can see detailed information of all the data that we have. And also you can get the amino acid sequences of every structure in a FASTA format. The limitation here is the PAE files are not yet available on the FTP, but we're working on that. Also, just to highlight, as part of the database, we do have frequently asked questions. So if you have questions like, how can I bulk download the data? How should I cite? And who should I contact with inquiries? We have all of these questions here in this tab. So AlphaFold database is a member of the 3D Beacons network. This is a collaboration between providers of different macromolecular structure models. And it includes AlphaFold, PDB-E, Model Archive, AlphaFill, Swiss Model, and others. This aims to provide model coordinates and meta information from all the contributing resources in a more standardized format on a unified platform. The data here is provided, is freely available for academic and commercial use. The molecules that you're going to find here, macromolecules that you're going to find here, that fall into 4 different categories. We have experimentally determined, that are based on data from techniques like crystallography, cryo-EM, NMR, and others. We have the template-based models. This is a Swiss model, is an example of the data providers that we have in this category. Then the ab initio models can use templates as an auxiliary input, but they do not depend on them. So this might sound familiar. And this is where you can find all the AlphaFold predicted structure in this category. Finally, we also have the conformational ensembles that are created using a combination of experimental data and computational modeling. And ensembles from the PED database are an example of this. And you can also view all the different models that we have for each category. This is how the entry pages look for 3D beacons. You can again inspect which parts of the amino acid sequence are covered by which models in a 2D viewer using the PD Prot Vista, which is this one. And you can display the structure using the same tool that we have in AlphaFold, which is called MolStar. And we can see here that the predicted structures from AlphaFold, they fall in the category under ab initio. Just to know that the main purpose of 3D beacons is to provide API endpoints for getting the models, although we do have a great graphical user interface. So just to wrap up the webinar, the main messages that I have for you is that there is a gap between experimentally determined proteins and protein sequences, although thanks to AlphaFold, we can now have the predicted structures available for research. There are over 214 million structures in the AlphaFold database, although the models have limitations, and it's very important for you to use the confidence metrics together whenever you're using the structures from AlphaFold. AlphaFold database is not the only way that you can access the structures. You can also access them all for the 3D Beacons network, and this will allow you to have a more comprehensive way to find structures. And thank you for listening to today's webinar, and a big thank you for everyone that's involved in AlphaFold database. So we are now open for questions, if there are any.
Okay. Thank you, Paulina, for today's webinar, and thanks a lot, everybody, for joining in. And actually, we have loads of questions, and thanks to David, who is here from PDBE, who has already answered several questions in there. But we still have many questions remaining, and we'll try to answer as many as possible because we have about 15 minutes. All right, so we'll start with the first. There's one simple question. What is the most important input for AlphaFold?
Well, there are actually two main important things for AlphaFold. The main one is obviously your input sequence, because if you don't give an input sequence for AlphaFold to predict, you won't be able to predict it. So I will say that, and the MSA is very, as a rule of thumb, whenever you're running AlphaFold, the MSA should have at least 30 sequences for it for you to get a reliable structure.
Thanks, Polly. And I think that answers several other questions which are actually about MSA. Okay, so the next question is, what is the AlphaFold prediction threshold for low scores in terms of percentage of amino acid sequence that justify a wet lab analysis like XRD or NMR structures? And can you elaborate on its power to predict IDPs, which is intrinsically disordered proteins?
Well, it's been found that whenever you see the PLDT that is lower than 50, it might highlight that this is an intrinsically disordered region, but not in all the cases, because it also depends on the environment of the protein. As a checkpoint of the predictions, the threshold for low scores. It is a difficult question to answer, because that will depend on what you're trying to do with the structure and what you think is a good prediction or not. So that will depend on your project.
Great, thank you. The other question, what is the comparator for the PLDDT scores? Does it compare the predicted fold to structures in PDB from closely related proteins in the MSA?
It's not actually using the true structure for the PLDT, just predicting how well the local environment for that residue is, if it's actually found as an helix. For closely related proteins, it does use the MSA as trying to predict that.
Is there a way to checkpoint AlphaFold predictions? For example, if you are running it on a server with resource limitations, can you do one prediction, stop it, and then resubmit your protein sequence and then do the remaining predictions?
You should be able to do that, but to do that, you will require some coding knowledge and change the code. So this is available, and it's on the CCBY license, which means that you can change the code for your needs.
Next question is, can you explain based on what algorithm the AlphaFold decides whether it takes template into account or use only MSA?
It will depend on the information that is taken from the MSA. So as a good thing to do when you are running your predictions is always check the MSA length and how many sequences you have there. One of the things you can also do is try to play around with MSA as an input and give AlphaFold a good amount of sequences and then a shallow MSA, and you can see how it predicts the structure differently based on that. So I think that also, goes into one of the questions that it says, is there a way to predict protein structure where the protein is free from ligand? Yes, there is a way for you to predict different structures, conformations of the same protein, and the way to do it will be to play around with MSA.
Right. Now there's a futuristic question. Will AlphaFold ever reach a good protein prediction for intrinsically disordered proteins? We already have many sequences for these proteins, but it's extremely challenging to get the structure for the monomeric proteins. What is your view on this?
I feel like this question it answers itself, because it's a disordered region or disordered protein. So for AlphaFold, it's difficult to know how to place those residues in that area of the protein.
Thank you. Is there a method to evaluate PAE in terms of total protein native structure that will help determine if a wet lab assay is needed? Probably about the validation of the structures.
The PAE, that will also go into, you're trying to use the whole protein or domains for the protein, because sometimes the PAE can tell you that the two domains are not in the right position in the structure for AlphaFold. So again, that will depend of like what kind of wet lab assay you're trying to do. I know we have so many questions, but I forgot to highlight, as part of the AlphaFold database, we do have a email if someone has more questions about how to use it. or the PAE, the PLDT, how to access. There is just one question that I'd like to answer to, and is the input sequence length limit for AlphaFold run. On top of my head, I think it's 2,500. So that'll be 2,500 amino acids for the input for AlphaFold to predict a structure.
All right.
I could just add to that if it is longer, AlphaFold will chop it into sections and then provide structures for overlapping sections of a longer sequence.
Yeah, that's right. Thanks, David.
Okay. There's another question, I think it's about cross-linking. So if we have cross-linking information between residues of a single protein, can we change or remodel this structure?
Can we change or remodel the structure? So this will be remodel the structure that you got from AlphaFold algorithm using the source code, I guess. Yes, you can play around with AlphaFold. You can try to give it templates also. And again, try to use a shallow MSA and see how that changes the prediction.
Next question is, I think, is about the integration of AlphaFold with UniProt, I think. Sometimes I find AlphaFold structure in UniProt to use for specific tasks. Can I delete the low quality part of the AlphaFold structure in this case?
You can't do it straight in the UniProt website, but you can.
Yeah. Are there any other ways to get a database and use it to look for a specific protein prediction? I know BigQuery is an option. Are there other such options?
Yeah, so you have the API. You also have the FTP to access the different versions of the database that we have. There's also the README file, so you can see the difference between all the versions and why we're now using the version 4 and when it was created too. You also have, as mentioned, the BigQuery. And the last one will be 3D Beacons network.
Now, there's one interesting question. I can see many versions of AlphaFold, like AlphaFold Multimer, CollabFold, HelixFold, OpenFold. Is AlphaFold still better in prediction in different types of protein families?
That will be AlphaFold. So AlphaFold Multimer is different. It's not the same as the AlphaFold that we have, because this is for single chains, and for the multimers it's different. CollabFold is the code from AlphaFold, but it's been tweaked, so it runs faster and it's a little bit better than AlphaFold. But I would recommend you to check the paper for that. So like every one of these ones will be, depends on what you're trying to do.
Thank you. How diverse should be the MSA? Is there any risk of including too many similar sequences on it or too many dissimilar sequences?
I feel like I'm repeating myself. Yes, the MSA is very important for AlphaFold. There's a risk when you use too many dissimilar sequences, because then it won't be able to actually get a sense of what is important from the MSA to predict the structure. So this depends what you're trying to do with AlphaFold. So if you do that, you might get low confidence values for the structure. But then if you include too many similar sequences and there's already a template that AlphaFold has seen, it's going to be very similar to that one.